Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine for a moment being able to precisely adjust, like a dimmer switch in the powerhouses of your cells, not just flicking them off, but subtly controlling a specific genetic message.
Right, fine-tuning it.
Exactly. And here's the kicker. Turning down one of these crucial energy units doesn't necessarily cause the whole system to just, crash immediately, which really flips some long-held ideas about how our cells handle stress on their head.
It absolutely does.
So that's the really electrifying discovery we're diving into today.
Yeah.
Our mission, essentially, is to explore this groundbreaking new tool that gives scientists unprecedented, almost surgical control over mitochondria, the cell's energy factories. We're digging into a fascinating article from Science that details this innovation, and we'll unpack what it really means for understanding health and, well, disease, too.
It really is a pivotal moment, I think, in cellular biology. Mitochondria are just fundamental, aren't they? To metabolism, energy. They underpin pretty much everything our bodies do. But for decades, trying to study their inner workings, like how individual mitochondrial genes are expressed, how they're regulated, that's been incredibly difficult.
A real black box.
Totally. And this lack of precise tools has been a huge roadblock, frankly, for understanding a lot of diseases linked to mitochondria.
Okay, so let's unpack this a bit for you, the listener.
Yeah.
Most of us remember mitochondria as the powerhouses from biology class,
The
right?
classic line.
The classic line. But what's often missed, and what makes them so unique, is they actually have their own little genome, totally separate from the main DNA in the cell's nucleus.
Tiny but mighty.
Exactly. And this mitochondrial DNA, it makes just a handful of super important proteins. Proteins essential for building this system called oxidative phosphorylation, or OXPHOS, basically the cell's electricity grid.
Precisely. And these OXPHOS complexes, they're these intricate molecular machines. But here's where it gets tricky. They're built using parts encoded by both the mitochondrial DNA and the nuclear DNA.
So a joint effort.
A real joint effort. Requires a lot of coordination. And the real hurdle has always been, well, how do you directly test what just one of those mitochondrial messenger RNAs, those specific messages, is actually doing inside a living human cell?
Right, without messing everything else up.
Exactly. The existing methods were often pretty blunt, manipulating nuclear genes that affect mitochondria or using harsh chemicals. It often disrupted the whole cell.
Like trying to fix a watch with a hammer.
That's a perfect analogy. Yeah, just not suited for the kind of precision needed.
Okay, so here's where this new study gets really ingenious. They developed something they call peptide-morpholino chimeras. Sounds complicated, I know.
It does sound like a mouthful.
But think of it like a custom-built microscopic delivery truck.
Okay.
Carrying a very specific targeted message inside.
And the elegance is really in the design, how it gets around those old problems.
Yeah.
They engineered these molecules called morpholinos, essentially. They're like tiny molecular blockers. Yeah, they bind to the RNA message. And they're super stable. They don't get chewed up by the cell's enzymes easily. Then they chemically linked these blockers to special peptides. Think of them as address labels or like an internal GPS.
Directing it right where it needs to go.
Precisely. Guiding the whole chimera across the cell membranes straight into the mitochondria and doing it without causing a lot of collateral damage.
And the really incredible part is just how precise and efficient it is. In their early tests using isolated mitochondria, they showed this chimera was way more effective at silencing a specific gene, MTCO1 in this case, than previous attempts.
Something like 5 times more efficient based on the IC50 value they reported.
Yeah, it's like upgrading from, I don't know, a clunky old TV remote where you have to mash the buttons to this super sleek, responsive dimmer switch. You can dial down that specific message with amazing control.
And a key question, naturally, is how does it actually work? What's the mechanism? They found the chimera works by basically sitting on the target mitochondrial mRNA. It physically blocks the ribosome, the protein-making machine, from latching onto and reading that message.
So it just stops the protein from being made right at the source.
Exactly. The instructions are there, but they can't be read.
Yeah.
And what's also really useful, they showed this isn't just a one-trick pony. The system can be designed to silence multiple mitochondrial mRNAs at the same time.
Oh, wow.
Yeah, which makes it incredibly versatile for studying how different mitochondrial components interact within that powerhouse.
Okay, that's amazing in isolated mitochondria.
Yeah,
But like you said, the real game changer, the acid test, is whether this dimmer switch works in living intact cells.
The crucial step.
Yeah.
And it does. They successfully got these chimeras into human HEK293T cells, and they saw a really robust inhibition, a slowdown of protein synthesis from mitochondrial genes like COX1 within just 3 hours.
2 hours, that's fast.
Very fast. And what's more, they could keep that effect going for up to 3 days.
3 days.
This kind of temporal control is just, it's vital. Think about it. You can add the chimera, see the immediate effect, then wash it out and see if things recover.
Right, so you can separate the initial hit from any long-term adaptations the cell might make.
Exactly. It gives you a much cleaner kinetic picture, sort of like watching cellular cause and effect in slow motion. And of course, they double-checked that the chimera really was going to the mitochondria, confirming its specific action.
Now, you might be sitting there wondering, okay, human lab cells, fine. But does this work in other, maybe more relevant cell types?
A very important question for applicability.
And the answer again is yes. They tested it in human heart muscle cells, cardiomyocytes, that they'd grown from induced pluripotent stem cells.
Wow, okay.
And also in mouse liver cells. And in both cases, they successfully blocked mitochondrial protein translation.
That's really promising. It shows the potential breadth of this tool, useful across different biological systems and questions.
Definitely. So, okay, you start dimming the lights in the powerhouse. What happens next? What kind of conversation does that start with the rest of the cell? Especially with the nucleus, the main control center.
Ah, the mitonuclear communication aspect. It's a fascinating area.
And this study gave us a really cool glimpse into that. They found that silencing these individual mitochondrial mRNAs significantly changed the nuclear genome's activity.
Meaning the nucleus started changing which of its own genes it was turning on or off in response.
Exactly, a direct response.
But what's really striking here is that it wasn't just some generic alarm signal. The pattern of those nuclear gene changes was specific to which mitochondrial message they silenced.
Oh, interesting. So silencing gene A in the mitochondria triggers a different response in the nucleus than silencing gene B.
Precisely. For instance, silencing subunits of complex I, like ND1 or ND4L, seemed to trigger broader cellular responses compared to hitting some other mitochondrial targets.
So it's not like the mitochondria just hit a big red panic button.
Yeah.
It's more like sending a specific coded message to the nucleus.
That's a great way to put it. It definitely challenges the older idea that maybe these responses are more generalized or, less specific. Their analysis, using some pretty sophisticated techniques, showed there were unique responses tied to silencing specific genes, but also some common pathways that lit up across different silencing events, things related to energy, metabolism, making proteins in the cytoplasm.
Okay.
It paints a picture of a much more layered, maybe even hierarchical communication system than we previously appreciated. The nucleus is really listening and interpreting specific signals.
That's fascinating. Okay, so we talked about the nucleus. What about at the level of the mitochondria themselves? If you dim a specific gene, say for complex I, what happens to the actual energy-producing machinery?
Right, down to the hardware level. They looked at that directly. And they saw a really robust and, importantly, selective decrease in the activity of the complex that they targeted.
Selective meaning.
Meaning, if they silenced a gene needed for complex 1, complex I activity dropped significantly, but the activity of complex 3 or complex 4 didn't necessarily take an immediate hit.
Wow, that is incredibly precise control at the functional level.
It really is. And it leads to this important, slightly counterintuitive question. If you knock down on these complexes, does the whole energy production system just collapse instantly?
Yeah, like pulling one Jenga block causes the whole tower to fall.
Exactly. But their detailed protein analysis, where they tracked protein levels over time after silencing COX1, that's a key part of complex 4, revealed something really interesting.
Yep.
The loss of the other complex 4 parts was gradual. It took time. And surprisingly, the other big OX-PHOS complexes, like I and 3, were not immediately or substantially affected in the early stages.
No way.
Yeah, it really challenges that immediate cascade failure assumption.
So the powerhouse is actually more resilient, maybe more modular than we thought. It doesn't all just crumble at once.
Precisely. It points towards a more organized, maybe sequential response to the stress. The study suggested that silencing COX-1 first hit its immediate partners.
Right.
Then it affected the proteins needed to assemble complex 4.
Okay.
And only later, maybe through broader signaling, did the effects start to ripple out more significantly to the other complexes.
So is a buffering capacity or maybe backup systems kicking in?
Something like that. Now, don't get me wrong, oxygen consumption, the main job, did decrease significantly, and the cells shifted towards glycolysis, a less efficient way of making energy.
So there were consequences.
Oh, absolutely. But interestingly, the mitochondrial membrane potential, which is a key sign of mitochondrial health, was largely maintained, at least for a while. It shows the cell has coping mechanisms, a surprising degree of resilience, even when one part is compromised.
That resilience aspect is really interesting. Now, this dimmer switch tool, it didn't just confirm things, right? Didn't it also help them find some new players involved in building mitochondria?
It did, yes. That's another powerful application. By seeing what changes when you perturb the system, you can identify proteins potentially involved in the response or in the process itself, mitochondrial biogenesis, as it's called.
And they highlighted a few.
They did. They pointed out 3 proteins, TM186, ZNF703, and SMIM26, whose levels changed quite a bit when they silenced TOX1.
Okay, what do they do, or what did we think they did?
Well, take TM186. It was known to be involved in building complex 1, but this study suggested its role might be broader, showing it interacts with parts needed for assembling complex 4 and V as well.
Expanding its job description.
Kind of, yeah.
Blah.
Similarly, ZNF703, which is known as an oncogene, a cancer-related gene,
Right.
they found it interacts with mitochondrial membrane components and seems to support the making of another protein, COX2, and SMIM26, this small membrane protein, looks like a really key organizer.
Oh, so?
It interacts with loads of factors crucial for building complex 4 and for quality control, even latching onto proteins as they're being newly made, these so-called nascent chains.
Wow. So this tool isn't just for turning things down. It's also helping us discover the hidden machinery, the construction crew, for the powerhouse itself.
Exactly. It's providing functional context, helping us place these proteins within the complex network of mitochondrial assembly and maintenance.
Okay, let's pull back a bit then. What does this all mean for the bigger picture, for science, for maybe future medicine? This precision dimmer switch feels like a really significant step.
Oh, it absolutely is. If you think about the broader context, this tool gives us a way to reversibly, programmably perturb mitochondrial gene expression. That's huge.
Reversible and programmable.
Yes. It complements other tools like CRISPR gene editing, which, has its own challenges when trying to target mitochondrial DNA directly. This offers temporal control, turning things off and potentially back on, and precision targeting inside living cells.
So it moves beyond the sort of sledgehammer approach we talked about earlier.
Precisely. It shifts the field away from what was often, by necessity, blunt force mitochondrial genetics towards a new era of precision perturbations. It aligns mitochondrial research with the kind of sophisticated systems biology analyses we can do elsewhere in the cell.
Which means researchers can now really put old ideas about mitochondrial diseases to the test, right?
Definitely. They can clarify cause and effect in ways that just weren't possible before, and potentially provide crucial insights for therapy. Imagine, maybe for some conditions, you don't need or want a permanent gene edit. Maybe a temporary partial dimming of a gene is actually a better strategy.
That's a really interesting therapeutic angle.
It is. This tool essentially provides a playbook, a general method, for systematically connecting specific mitochondrial genes to their functions and downstream effects, all within the natural environment of the cell.
So this deep dive has really shown us how a cleverly designed molecule, this chimera, can unlock whole new levels of understanding about our cellular powerhouses.
Absolutely.
We now have this dimmer switch for specific mitochondrial messages. And using it has revealed this more nuanced, almost hierarchical conversation between the mitochondria and the nucleus. And it's powerfully challenging some of our basic assumptions about how these vital organelles respond when things go slightly wrong.
It really forces us to rethink the dynamics. And this perhaps raises an important question for you, our listener, to think about. Now that we have this kind of precision tool, What other hidden sequences of events, what other really subtle signals inside the mitochondria might we finally be able to illuminate? What new mysteries in that complex dance of cellular life and disease might this dimmer switch help us unlock next?
That's a great question to ponder. Thank you for joining us on this deep dive into the really fascinating world of mitochondrial science. We hope you feel a little more clued in and maybe just a little more curious about the incredible complexity humming away inside your own cells.